Nematics. Thank you, Rodrigo.
Okay. Thank you, Pablo, for the introduction, and thank you the organization for this amazing event. I think it's, for me, that's starting now in this position. It's great to know people in Brazil, Latin America, and other parts of the world, USA, Europe, working in the same topic. I think it's a great initiative to build this community. Okay, so this work was done in collaboration with an experimental group at the University of Barcelona. I will present, but yeah, I have a first slide just to let you know which is my expertise, which kind of topics do I work. So have you been working with fluid dynamics, mainly in fluid simulations, in topics like fluid flows in porous media. In this case, it was inspired by the food industry, like the problems like the coffee extraction, how the fluid flows in time-evolving porous media. I have also worked with droplets and capsules and flowing particles in fluids. In liquid crystal kernels, especially in hydrodynamic effects in liquid crystal kernels, I will present a talk on Friday. If you are interested, you can call. We can go. Is the last one, and in active nematics. So all those topics they involve fluid simulation, more specifically using the same techniques that's the lattice bolts. That's the tool that I have been working since my undergraduate degree until now. So this is also to let you know that I'll show main experimental results, as I told you, but my contribution was in the simulation part. I actually don't know how to use a microscope. It's not that I don't want to, but I didn't have this training, unfortunately. So, well, to start with, you probably have seen, well, we have seen yesterday and today with Chantal, some active Brownian particles, and so if the active matter in general, well, to start with, is made of particles that is able to extract energy and convert into motion, right? So if those particles, they are scattered, you have low density of these active Brownian particles, they behave almost individually. So you have an individual behavior. One bacteria wants to go one direction, and rarely they interact with the others. While when you have higher density of bacteria in special conditions, they develop the swarming mode. So they start to behave collectively. If you freeze this image, you see that most of the particles, most of the bacteria, they are aligned in the same direction. So they acquire a nematic order. So, and also, as I'll talk in the next slides, if you analyze the velocity field, it resembles quite a lot fluid turbulence, but it occurs at low Reynolds number. So you have a kind of turbulence called active turbulence, but at small scales, so at the scales of nanometers or micrometers. So, and active nematics is formed by dense populations of those elongated particles, like in colonies of bacteria. And as you can see here, most of the particles for one specific region, they are aligned with each other, which resembles what happens in passive liquid crystals, in the nematic phase of the liquid crystal. So the liquid crystals, the liquid crystals can be, well, in the solid state, so with ordered and rigid positions of the particles. That can be in the isotropic phase, where all the particles, they point in random directions. And in between, for intermediate temperatures, the liquid crystals, they form the nematic phase, which can still flows like a fluid, but it's ordered. So I'll talk about the nematic, this analogy with the nematic phase of a liquid crystal. So one example, as it's here, it's shown here, are colonies of bacteria. Another very interesting example are mixtures of microtubule and kinesin. So those components can be found inside the cells. The microtubule gives the rigidity of the cells and also form the flagella and the cilia of bacteria, for instance, while the micromotor is the kinesin, transport the organelles from one side to the other of the cell. So so it basically uses the microtubule as a guide for the transportation. So a Dogic group, the Dogic and his group, had the very nice idea to put only those two components at the interface between water and oil. So you have effectively a two-dimensional system, as it is illustrated here. Where the microtubule and the kinesin, they deplete to this interface, and you have the formation of active turbulence at 2 dimensions. So basically, the kinesin makes the microtubule to shear with each other. So you have a dense concentration of those two components. You have again something that resembles turbulence. So it's like a chaotic flow where it's not bounded. So it's again active turbulence, but in a different system. So those flows, they are driven by mainly by the positive defects, which are comet-like. The topological charge of these defects, they are placed 1/2. It's basically the line integral of this director field, while the negative defects, they are just dragged by the positive defects. So they are not self-motile. So here you can see some static images of those defects, the comet-like and the trefoil-like defect. In the simulations and experiments, actually the continuous simulations, the continuum model for liquid crystals describe quite well this mixture of microtubule and kinesin. And here you see an illustration that those defects, they can be created from zero topological charge. So you have initially aligned particles only, and those two topological charges, they can be created, and when they meet again, they are annihilated. So you have all the time during the active turbulence this creation and annihilation of topological defects. We always have this characteristic length for the active nematics, which is a competition between the distortions. We have penalization for these distortions in the liquid crystal or the active nematics. And it's a competition between this free energy and the activity that tends to distort more. So from this, you have this characteristic length, it's called the active length, given by this square root of 1 over the activity, which is the zeta parameter. So yeah, those defects, they can be found in many other systems in 2 dimensions or even in 3 dimensions. Like in our fingers, you can observe some topological defects because you have those lines in 2 dimensions. So you can observe place on 1/2, like those comet-like here. You can observe minus 1/2, these trefoil ones, also place 1. And you can observe in other systems, like even in our hair, you can see these defects like a vortex, because you cannot align all the hair in one direction. It would be a bit weird. So actually I should avoid this comparison, because the last time I presented was full of bald people in the audience. So they got a bit annoyed with this. So, yeah. So many other systems can be described by these models with active nematics, like in growing colonies of bacteria, epithelial tissues, epithelial cells, that they are usually isotropic, but when they are moving, they develop those effects in the nematic order as well. Other kinds of cells, like the stem cells and the fibroblasts, and so on. Okay, and as I mentioned, those colonies of bacteria or microtubule kinesin, they develop chaotic flows called the active turbulence, and it occurs for small Reynolds numbers. So it's interesting, for instance, for mixing applications that I will mention in the end, because mixing is difficult to produce at small scales or at Stokes regime. Usually use fluid turbulence to mix fluids, like you use a blender to mix even water and oil, right? You produce vortices. But at the small scales, it's more difficult because the Stokes flows they are reversible. So it can be an option to use those mixtures of microtubule kinesin to mix fluid in microfluidic applications. And in living systems, this mixing, they are already used to mix nutrients, for instance, to spread the nutrients over the colonies. So the spectrum of this active turbulence is still under debate, so actually it depends on the kind of system. For bacterial colonies, we usually observe these scales, these two ones, 530s and minus 830. It was reported, for instance, in this experimental study using Serratia marcensis. And we have been able to recover this in a previous work using computer simulations with active nematics. So, but there are other possible scales, as can be seen in the review of Ricard Alert, for instance, in active turbulence. Since this mixture of microtubule kinesin, people already know for quite a long time, don't know, 10 years or a bit more, and people are thinking applications for that because it's relatively cheap to produce, it's easy to control, and it's very reproducible. If you follow if you follow some steps, we always find this kind of flow. So one possibility was given 2 years ago by the Dodge, who invented this system, to produce micromotors. So similar to the ones Chantal have presented, but here there are differences from other works because this rotation that was found is produced by topological defects that are placed behind each asymmetry and makes this rotor to rotate. So this can be used as a rotor in 2 dimensions in this kind of setup. So one possibility. There are other proposals to produce logical operators. But if you want to use in true microfluidic applications, we want to pump fluids. So we know how to pump fluids in 3D. We basically can impose a force and make the fluid flow like gravity. Right? You can have a pressure difference between one side and the other. But if you do this with this system that is 2-dimensional, you mix everything. So you no longer have the interface between water and water with the active nematics in between. So how to pump fluids in truly 2D systems or quasi 2D systems like this one? So that's one of the questions we will address here. Okay. So, I forgot to control the time. You can let me know when it's close. So to describe this, we use the continuous simulations. And it means that we are using a mean field, we are using a mean field model. It means that we are not considering individual particles, like we are not simulating molecular dynamics or things like this. It's a continuum that we consider the average of those particles, and we use this tensor order parameter. This is to have a head-tail symmetry of those particles. We are only interested in the direction of those particles, not the vector. And so we have here this scalar order parameter that gives how well the particles are aligned with the main direction, and n is the main direction. Okay, those particles, they align with the flow in a certain way that's given by the continuous theory. So more elongated particles, they align more with shear flows, while spherical particles, they don't align at all. So it's related to the shape of the particles. There is a penalization of distortions, like the band distortion and the display one. And most importantly, we have this active stress that gives the motion of these active nematics. So I have here the activity parameter, zeta, and the tensor order parameter. And giving a bit more details, so we solve 2 equations which are coupled. The first one is the Beres-Edwards equation. It gives the time evolution of the tensor order parameter. So this equation depends on the fluid velocity here, and also in this co-rotational term that gives how the particles rotate with the flow. And the Navier-Stokes equation, which gives the time evolution of the velocity field. And it also depends on the tensor order parameter that's given by the previous equation. So those two equations, they are coupled. And here we use a simplification for those that are used to simulate active nematics. We are not considering here the passive stresses, because it was already shown in experiment in simulations that typically the passive stresses, they are much smaller than the active ones. So 2 orders of magnitude. So we can neglect for simplicity. So we are considering here the active stress and this coupling between the two equations. So okay, so we have tried a few different configurations, and we saw using simulations that when you use an array of triangles, we can direct the flow, which is usually chaotic of these active nematics. So here you can see the director field. It's not so easy to observe that it's flowing to the right, but you can see this in the velocity field. And this group in Barcelona, led by Francesc Sagüez and Jordi Ignés, they come with a technique, a new technique that was published last year, to solidify regions in situ. So they create obstacles in situ. For instance, here you have initially chaotic flow, and they just shed light in a certain region, and this region becomes attached to the substrate. So you have a three-dimensional system with active nematics in between, and you have a column of this material that is polymerized. So you have a polymerization of this fluid, and this obstacle can be more flexible or more rigid, depends on the chemistry. So it depends on how many monomers you have, the intensity of the light, and so on. So in this case, it's a flexible one. But one thing that we will explore in the end is that this technique can produce changes on the fly in microfluidic circuits. So that's something that's not possible in usual microfluidic experiments. So yeah, so they have their initially chaotic flow, and then they create these lattice of triangles, and they observe the chaotic flows the directed flow that I will show in the next slide. So here we have some images of the director field in simulations and experiments. The velocity field, you can see here time averages. So you already have a certain duration where we average this. And you observe that these red spots between the triangles, the obstacles, both in simulations and experiments. And we can also observe some vortices that appear behind each triangle. So we believe that those vortices, they help to direct the flow. So they can be better seen in the vorticity field. You have two vortices with opposite directions behind each obstacle, and it helps the flow to go to the right. So both in the experiment and simulation. Well, this kind of system was already studied in other systems. It was observed that we can observe directed flow in Brownian particles or active Brownian particles using headset systems similar to those ones. But here what is different is again the mechanism behind this. So we have hydrodynamics, we have a pressure buildup that I will show later. We have the formation of those vortices, and also you have the topology helping to direct the flow. Because here you see the director field. There is both in experiment and simulation, those topological defects, the positive ones, that drives the fluid to the right. So we believe the shape of the triangles help to produce a topological defect here between the defects. So topology and hydrodynamics, that's the difference from other headset systems. Okay. So we characterized this system first using simulations, and then the experiment confirmed it. For instance, which is the best size for those triangles. So keeping the cell size constant, we use a different triangle size here in the x-axis. And we observed that the velocity is always positive, so it's always going to the right. However, it increases initially with the size of the obstacle, and then it starts to decrease slowly. So there is an optimal size for the triangles to pump the fluid. And interesting, this optimal size is related to the characteristic size of the active nematics that I mentioned before. So they are related to the average distance between the defects. So the optimal occurs exactly for an obstacle size that is close to this characteristic length. And it makes some sense because that's the size that accommodates perfectly here a topological defect that I have shown before. So we later changed the cell size and also for different triangle size, and found here an optimal configuration that we kept constant in the next results that I will show. Okay, so here is we see some experiments for different numbers of triangles, isolated triangles, and it was observed that the pumping effect can only be observed when you have at least 4 triangles. So we have these unit cell for a pumping system. That's because we need the formation of those vortices behind the triangles. And here there is no clear preferential orientation for the flow, easy flow, because, well, you have 3 directions given by the edges. Here you start to have something, but it's not so clear, and you need at least 4 to have a unit pump. So what happens when you have disorder in your LEDs? Because in an experiment, it's not so easy to make extremely irregular LEDs of triangles, squared LEDs. So using simulations, we saw that when we start with squared LEDs of triangles, and we start to displace those triangles with a certain Gaussian distribution that's given here by the standard deviation. When you increase this disorder, the average flow decreases. So we do need another squared LEDs to have the pumping effect. If you have a disordered one like this one, the flow is, doesn't have a clear, easy direction. And the obstacles can also be flexible. So this we explored only using simulations. Actually, asked a student that was working with a similar topic to test this, to try to replicate these images at least, and to put triangles that could move around a certain center of mass. So here we have more rigid triangles, and here they can move around a certain center of oscillation. So when we increase the rigidity of this spring that attaches the obstacle to the center, the velocity increases. So again, we need some rigid obstacle to guide the flow. And if you have more flexible triangles that could distort, and here you have more rigid and here more flexible, when we increase the rigidity again, it pumps more. So the more rigid we can produce the triangles experimentally, the better for pumping, for the pumping effect. And if you are considering pumps, like in hydraulic engineering, like when we want to extract oil, for instance, we need very high pressure. So what we do is, I don't do this, but when we extract the oil, we need many pumps in series. So we couple like dozens of pumps in series to have very high pressure. So the pumps in series, they have this characteristic to increase the pressure between one side and the other, while keeping the flow rate constant. So that's what happened for ideal pumps, ideal hydraulic pumps. So we can test this in our case. Does this system follow the ideal law for a combination of pumps? So what we do is to change this, the number of columns that is analogous to have pumps in series. So the velocity profile across different numbers of triangles, here we have 15 blue, 5 in red. So the velocity have those spots between the defects, between the obstacle that you have seen before, and at some point it starts to saturate. And here you have the pressure along this system, and we also have some peaks, but this increases more linearly. So pressure is something that we don't have with usual Brownian particles, for instance. We don't have hydrodynamics here. And so we can relate this pressure difference between one side and the other of the pumps, and also the exit velocity. And when we plot this against the number of pumps, we see that initially increases, and then it saturates. That's the behavior that you would expect for ideal pumps. The velocity field or the flow rate keeps constant with the number of points. So after 10, it follows the rules of ideal hydraulic pumps. And for the pressure field, pressure difference, it increases more or less linearly. That's close to linear. That's what we would expect for ideal pumps. So after 10 pumps in series, it follows the laws of ideal pumps. Okay, and for pumps in parallel, what you would expect for hydraulic pumps is that the flow rate keeps increase with the number of pumps, while the pressure difference remains constant. So that's what we observe, that the flow rate increases with the number of pumps, more or less linear, while the pressure difference remains more or less constant. So for the combination in parallel, it's closer to what we would expect for ideal pumps. Okay, so since we can pump fluids, we want to pump in somewhere. Like we use it here an example for pumping inside the channel, a certain channel, both in experiment and simulations. And there is this flow profile that's similar in the experiment and simulations. We have those spots that are given here with higher velocity between the obstacles, and then inside the channel it starts to decrease. And it depends on the separation between the plates, right? How this decrease occurs. So here we measure the velocity inside the channel for different separation between the plates, and compare it with the flow field, or the average flow field, without the triangles. So without them, here in red, the average is always close to 0. Right? Because the flow field can go to the right or to the left. There is nothing that breaks the symmetry. We might have directed flow during some time, but it can even revert along some time. While with the with the triangles, we always have positive velocity. It's always above 0. Right? And we can also think in more elaborated circuits, microfluid circuits, like we don't need to pump only one direction. It can change. And we will not follow the full video, but the particles do have a positive circulation. So if you calculate the circulation of this flow field, it is rotating clockwise. They also tried this in the experiments. And as I mentioned before, one interesting feature of this experimental technique done by my collaborators in Barcelona is that we can change the configuration, the obstacles in situ. So, for instance, if we start with small triangles that pump the fluid in this direction to the right, we can later reverse the flow by placing larger triangles in the other direction, like in this case. So they both overlap, and the previous one disappear, and then the flow field start to go to the left. So we can observe this in the average velocity. In the small triangles, the flow field is not so ordered, but is always positive, so it can oscillate a bit more. And after the creation of this second of those larger triangles, the flow field is inverted, so it goes to the other side. So they also tested this with experiments, but they don't have here the video to show. You have to believe me. Okay. And so we can pump fluids. What else can we do? We can mix fluids, solids, like at the micro scale. You probably have seen these many times. Most of you have seen this mixing experiment where we put different colors of droplets in a very viscous fluid, rotate the cylinder in one direction. It seems to be mixed. And then when we revert the rotation, the droplets return to their initial position. So it's rotating. So now it's coming back to the original one. And you observe that actually the three droplets of different colors, they are not really mixed. So this illustrates the reversibility of the Stokes flows, because the Stokes equation does not depend on time, and it's linear in the velocity. So if you sum one solution or one motion in one direction, and then the opposite direction, it returns to the initial one. It does not happen to the full Navier-Stokes equation due to the inertial terms, but with the Stokes regime, you have this. And here it's illustrated at the macro scale with a very viscous fluid, still in the Stokes regime, but it also happens for normal fluids like water and oil at the micro scale. So it happens, for instance, for bacterial flows or those active turbulence in microtubule kinesin. So we tested different configurations for mixing solids. So we want to mix those two droplets, red and blue. And when we use these isotropic lets of obstacles, which are circular, we do have mixing. At the end, they will be very well mixed, but the center of mass remains fixed. So it's more or less constant. While with the triangles, we have something interesting that it can still mix in the vertical direction here, because we have quite strong flows in those directions that I will show in the next slide. And we also have downstream. That's something that we would like to have in microfluidic applications. We don't want to mix only two droplets of fluid. We want to inject. We have a constant flow rate of two solids, and at the end, we want to collect this mixture on the other side. So this kind of setup could be used for this purpose. And it's interesting that this system of microtubule kinesin is biocompatible, and it could be used for pharmaceutical or I don't know, living systems. So here we see the directions of the traces. They mostly go to the right, both in experiment and simulations. We see that often they change the channel, like we call channel this space between different lines, and sometimes they get trapped by those vortices that were here. And when we analyze the time that each tracer stay in the same channel, most of them we observe an exponential decay. So most of the traces, they stay very shortly in the same channel. And the probability of staying in the same channel decays with time. So it means that it's a good system for mixing in this vertical direction, the transverse to the flow, to the mainstream. Okay, to conclude, we have presented what is active nematic, and also how to direct the flows, which are usually chaotic, using a lattice of triangles. Those systems, they behave like ideal pumps at least after 10 pumps. And it could be used for pumping fluids in 2D. That's something that's not so easy to do in microfluidics, and could be helpful, for instance, in when you have colloids that sediment at the interface between water and oil. You might want to avoid crystallization of those colloids, or mixing surfactants and things like this. And yeah, so it's also a good system to mix fluids and produce downstream at the same time. So here I acknowledge my collaborators, the people that performed the experiment at the University of Barcelona, Jordi, Francesca, and Ignacy. My collaborators at the University of Lisbon, Margarida and Nuno, and the students that work with me on these and in similar topics involving active nematics. So that's it. Thank you for your attention. I'm open for questions. Yeah.
That's great, Rodrigo. Thank you very much. Danilo?
Thank you. Very interesting simulations. Can you visualize the topological defects and shed light perhaps into the mechanism behind the pumping, for instance? Well, what is happening with the dynamics of the topological defects that's causing this flow? Yeah.
The dynamics. Well, we didn't track the topological defects. So this image that I have shown is for a time average. Where was it? Is here. So we took a time average of director flow and observed that these there are those positive defects. So they are usually moving in this direction, the comet-like direction, but we didn't track them. And here we can also observe those positive defects behind the obstacle that are those ones that drive the vortices. But we didn't track them.
I see. I remember seeing a talk or a lecture by Julia Yeomans, yeah. And she describes everything in terms of the dynamics of the topological defects, and I wonder what it would do for flow in general for this kind of thing. Yeah. Anyway.
Actually, the whole flow, they are driven by those positive defects. So that are those that are moving, because those negative ones, they are just being dragged. So we can also observe that those negative ones, they stay in the region with a smaller velocity, like here behind the obstacle, or between remember that the higher velocity are here, right? So they stay here between the two triangles. So they are static, or they are just passive obstacles, while the positive one, they are moving all the time.
Just a quick comment. If you have active Brownian particles, right?
Yes.
These particles, they will spend less time to overcome the obstacle if they are going to the right, in this case, than if they are going to the left,
yes.
Right? So this is what originates the flow, right?
For Brownian particles, yes, but here
for active Brownian particles, yeah.
For active Brownian, and not even Brownian particles, they could go easier to one direction than to the other. So because one particle could, when they come from this direction, it could be directed toward the center, while in this direction it just hit like a wall. So yeah. It's easier to go in one direction. But here we have collective effect. We have the hydrodynamics, and also the formation of those vortices, the positive effect that also help to drive the flow.
But in the passive case, this is very small effect, right?
Yes.
In the sense that particles are not persistent, so they just...
Yes.
Yeah, okay. There.
Please. So, thank you for the very nice talk, Rodrigo. I was wondering, when you showed us the energy spectrum for the turbulence which is produced by bacteria, whether there is the analog of some kind of cascade, or perhaps an inverse cascade, or...
Let me come back to this slide. Yes, this is a complicated topic. There is a lot of debate. People don't agree if there is a cascade or not, but making an analogy with passive turbulence, we do have a cascade of a larger vortex being breaking into smaller vortices and so on until they reach this viscous dissipation and then they disappear. So we have this decaying cascade with other power law. It's actually minus 530, and so in passive systems, in passive turbulence, we do have this cascade, the decay one. This increasing region, it's something characteristic from the active systems because we have the injection of the energy into the system. So it starts to inject here. So at a smaller wave number, or so at largest scales, and then it goes to smaller scales and decay. So there is a lot of debate. I will not say that there is for sure a cascade, an energy cascade, but there are some power laws that seems to occur for bacterial turbulence. So we usually observe those two power laws, but for microtubule kinesin, actually they are others. They are 1, -1, and -4, if I'm not wrong. So
so there's no universality.
No.
Okay. Thank you.
Not that I know at least.
So I had a follow-up to Danilo's question. If we can go back to the triangles, I take it you're using planar anchoring conditions for the director field. I think that makes each triangle equivalent to a -1/2 defect. And so aren't you thus requiring extra +1/2 defects in the director field? And can you quantify how much extra activity that gives you?
Yes. So it's true. I'm using the planar anchoring to mimic the experiment, and indeed you have well. What happens is that you still have. I don't know if it will affect the flow, but you still have director field inside the obstacle because you initialize the system with a turbulent state, which is more or less isotropic. Well, we have the same proportion of negative and positive defects, and then you solidify this region. I try to do the same with the simulation as well. So although you cannot see, there is director field also inside the obstacle. And I actually try I measured the number of defects, the positive and negative ones, and they were the same. So it might be because if you have extra defects, I don't know, they don't annihilate. I think it's because they are inside the obstacle. But I measured, and they are more or less in the same proportion, with a very small difference between the number of defects of different signs. But yeah, it's a good question. If it's induce more flows, because we have more positive effect.
Thank you for the talk. Do you compute the mean square displacement when you have these triangle obstacles?
Sorry, could you repeat? I was looking to.
Yes.
Okay.
Do you compute that when you have these particles inside in these triangle obstacles? Do you compute the mean square displacement?
Mean square displacement of the tracer particles.
Yes.
Yes, I did. I just don't put here in the presentation. I don't have an extra slide for that. Well, in average, they have a linear. Well, the mean square displacement follows a Balisk law. So because the particles they sometimes get trapped, they move in the vertical direction as well. But in average, they are moving with a constant velocity. So if you take the center of mass, it's moving with a constant velocity. And if you take the MSD with respect to the center of mass, they are basically diffusing. So yes, I did. I just don't have here to show.
Are there differences between this when you have these circle obstacles and
the
other triangle obstacles? Are there differences?
I didn't remember if I have measured for the circular, but yeah, I would expect that they are just diffusive. But I'm not sure. I just suspect.
Thank you for your nice talk. I was wondering about, you thought that there is an optimal size for these obstacles, triangles. I was wondering if you have a check if the simulation box has any effect on this optimal size or no?
Yes, good question. So yes, for this figure, we have kept constant the cell size and changed only the obstacle side, the triangle side. So we saw this optimal size for this cell size. So and here in this second one, maybe I was quick, but we also changed the cell size, which is called here the L. So we changed this ratio between H and L, and well, for this one, well, this figure corresponds to this line, the half 0.5, and we found this maximum velocity for this configuration. But for the other ones, you can also observe something similar. For instance, for this one, you have a higher velocity here in the middle, but the optimal triangle size might change with the cell size. So we have something similar for other cell sizes as well.
And if you use the periodic boundary condition,
Yes.
here you used a periodic boundary condition?
Yes. Yeah. So for this study specifically, we only used one cell with periodic boundary conditions, and for the others, we changed this. For instance, here we also have periodic, but more triangles. In other cases, we have a closed box with a larger domain, just to have something similar to the experiment. We changed the periodic, the boundary condition depending on the situation. For instance, when we studied for instance, here we used a closed box, which a domain that's large, so it's not shown the entire domain here. And for the here you can see better. For the channels, we see that the velocity is 0 around. So here I'm really showing the entire system. So the boundary condition changes according to the setup that you want to study.
And another question, why you choose the triangle? Why you don't choose any other shape for this obstacle?
It's just because it was simpler, and the simpler asymmetric shape that we could think. But indeed, we could think in something like an asteroid. So that when we tried these flexible triangles, they actually acquire these more curved. It seems to be an equilibrium shape. So we could start from something that is more curved. But yeah, it's just to have something simple and with an axisymmetric shape. Yeah, but could be others, yeah. Could be more elongated triangles, could be
Ellipsoids also.
Half ellipsoid, yeah. If they half circles, yeah.
Half circles,
Yeah.
that facilitate the movement of the particles.
Yes.
Okay, thank you so much.
Very nice talk. Can I ask a quick question? Can you show the microfluidic device with the channel?
With the channel.
Yes. I was wondering, would it be possible to combine the two geometries, meaning a channel with funnels? Because it is known that a channel with triangles inside is actually a tool to actually pump bacteria, run-ensemble particles from one side to the other.
Yes.
So if you just combine this smooth channel with, triangles on both sides, you can, maybe you can gain the most.
Sure, it breaks the symmetry, so and direct the flow in the same way. So actually, we tried this. Actually, the experimentalist George, they tried this in the experiment. It does pump the fluid in one direction using active nematics as well, like the bacteria.
Exactly.
Yeah.
Okay.
So it occurs.
Thanks.
So what's the dependence between the rectification velocity and the level of activity?
Let me back to this slide. Good question. So here we tried for different activities, and the curves scale with a characteristic velocity, but the velocity it scales with with a square root of the activity. So we used this already in the scaling. So yeah, the larger the activity, the larger the velocity, but it follows a square root.
Square roots. Yeah. Okay. So if you have a mixture of active pneumatic fluids with different levels of activity, then keeping average the level of activity and increasing the diversity of activities, you will increase or change the level of rectification rate.
Yes, for sure. Maybe we could have other effects. It's a good system to try, but we could have something like the MIPS as well. You could have other things. I don't know if they will remain mixed homogeneously with time. So maybe they segregate somehow.
Yeah. We have done this for ABPs. We can talk about this.
Okay, good.
Just a curiosity, did you use some kind of package to solve the equations, or did you write your own?
No, I wrote my own code. Actually, I'm doing this for quite a long time. So just in Lisbon, I was for 7 years. So in the beginning, it was was difficult. It took 6 months to write this code, and kept improving. But I had already experience in the Let's Bootman method for other applications. So before I had worked in industry, my PhD I used the Let's Bootman for studying hydrodynamics effects in electrons. So so I just changed the system, and well, this equation is quite complicated to implement, at least the full one with the passive ones that I also have. But yeah, I implemented myself in C++.
Very nice.
Okay, so if there are no more questions, let's thank Rodrigo again.
Thank you.
And now it's time for coffee break.